Thank you. I'm glad to be here and present my master degree project on the role of substrate-borne vibrations in aphids' interaction with natural enemies. Okay, the subjects of our study are Myzus persicae, the aphid Myzus persicae, which is an agricultural and worldwide key pest and an efficient plant pathogen vector. And it's to natural enemies available for biological control, the ladybug larvae Adalia bipunctata and the endoparasitoid and Braconidae Aphidius colemani. And we decided to deepen our knowledge on their interaction with this aphid species because very little is known about their modes of interaction and communication. And also the role of vibration in aphids' interaction with these two natural enemies in particular, but also in general, is unknown. And so we wanted to answer to these two questions that you can also read. So if aphids and their antagonists in some ways communicate or interact through vibrations, in particular substrate-borne vibrations, and if it's possible to interfere with aphids' behavior, reproducing this, the antagonist vibrational cues. So, deepening our knowledge on the modes of interactions between aphids and their antagonists could in some ways pave the way for studies about new sustainable control tools that could be bio-inspired by the way insects communicate through vibrations, as it has already been done with other K-pest species or families, like, for example, leafhoppers or stinkbugs, for example. So the aim of our study was first to record and characterize the vibrational cues produced by aphids and their antagonists with the laser vibrometry technique. And two systems were taken into consideration both for Adalia bipunctata larvae and Aphidius colemani. The reaction of aphids were studied in presence of just one adult female of Myzus persicae and one adult female of Myzus persicae and a variable number of nymphs from 1 to 3. And the systems were at the same time also filmed with a camera. The second purpose of our study, and it's just a little bit more applied, so we wanted to assess whether the reproduction of the vibrational cues produced both by aphids and antagonists could in some ways affect the feeding behavior of aphids using the electrical penetration graph technique in a playback experiment. So here's some preliminary observations on about the behavior of aphids in presence of Adalia bipunctata larvae. You can see that Myzus persicae tends to react mostly when the larva starts chewing an individual, both an adult or a nymph, and their reaction is of defense or escape. In the case of Adalia bipunctata, Myzus persicae tends to escape more than react in a defensive way, like they walk away or drop off the plant. While reactions to walking, so the walking vibration or touching of Adalia bipunctata are less frequent. In a Aphidius colemani experiment, so far we observed just one reaction. Okay, the number of replicates is still really small, so Myzus persicae reacted mostly after several oviposition attempts made by the Aphidius colemani adult female approaching. And so these are just preliminary observations about their behavior. And okay, this is my last slide. We want to isolate the vibrations that in some ways caused a macroscopic reaction in aphids' behavior, so defensive reaction or an escape reaction. But we would like to assess whether also those vibrations that apparently didn't show a macroscopic response, for example, the ovipositor touching plant during us searching by Aphidius colemani female, could also affect aphids' feeding behavior using the PG technique. And that's all. I would like to thank my supervisor and for their mentorship and you for your attention.